In this lecture, we are going to focus on the entry point and life cycle of Sars Cov2. As a transmembrane protein, ACE2, which is angiotensin converting enzyme 2, serves as the main entry point into cells for some coronaviruses, including HCoV-NL63, SARS-CoV, the virus that causes SARS, and SARS-CoV2, the virus that causes COVID-19. More specifically, the binding of the spike S1 protein of SARS-CoV and SARS-CoV2 to the enzymatic domain of ACE2 on the surface of cells result in endocytosis and translocation of both the virus and the enzyme into endosomes located within the cells. Let's have a look at the location of ACE2 receptors within the human body. ACE2 is present in most organs. ACE2 is attached to the cell membrane of mainly lung type 2 alveolar cells, enterocytes of small intestine, arterial and venous endothelial cells, and arterial smooth muscle cells in most organs. ACE2 mRNA expression is also found in cerebral cortex, hypothalamus, and brainstem. The expression of ACE2 in cortical neurons and glia makes them susceptible to a SARS-CoV-2 attack. Now I am going to talk about the furin enzyme. It is an enzyme in humans that is encoded by the furin gene. Now, what is the function of this furin protein or enzyme? This furin protein or enzyme has a proteolytic activity, which converts a protein or an inactive protein and modifies it so that it becomes functionally active. This furin cleaves and modifies certain portion of the S protein, thereby making it functionally active so that it can bind to the receptor. The receptor binding domain constantly switches between a standing up position for receptor binding and a lying down position for immune evasion. Now, the diagram depicted here is the standing up position. Moreover, to fuse membranes, SARS-CoV spike needs to be proteolytically activated at the S1-S2 boundary, such that the S1 dissociates and S2 undergoes a dramatical structural change. These changes are brought about by the furin protein. The modified S-glycoprotein can interact with the cell surface receptor ACE2 angiotensin 1 converting enzyme 2. The S1 subunit, which contains the receptor binding domain, makes contact with the ACE2, which is facilitated by furin cleavage. Recent studies have found that S protein of SARS-CoV-2 is between 10 and 20 times more likely to bind to human ACE2 than the S protein of the early 2000 SARS-CoV strain. The heightened affinity for a prevalent cellular receptor may be a factor which increases transmission. Now that we know how the spike protein interacts with the ACE2 receptor, we'll now discuss about the viral life cycle. As soon as the SARS-CoV-2 virus and ACE2 receptor come close to each other, the virus is internalized via endocytosis. On reaching the cytosol, it begins to uncoat and release its genetic material. The positive sense single-stranded RNA, with the help of host ribosome, translates RNA polymerase. Since RNA polymerase is used for transcription, it transcribes negative sense single-stranded RNA. This negative sense ssRNA in turn produces single-stranded RNA molecule, which is positive sense. This step is repeated several times to produce large amounts of RNA, part of which becomes the genetic material, and the remaining part encodes other viral proteins. Together, they are packaged in the Golgi body and they are released into the cytosol. Once the viral load increases, these viruses move out of the cell through exocytosis. If you like this video, please like, share, comment and subscribe to this channel. If you want me to make a video on any particular topic, please write it below in the comment section. Thank you.